me know if you are able to see my screen. Oops. Anyways.
We can see it.
Okay, awesome. Okay, so you can see question number 1. And I would like you to start answering those questions. Okay. The first question is asking you about, and I figured out that probably previous lectures are already kind of 2-3 weeks ago, so probably you start forgetting them. But I want you to also remember that some of these lectures are really important, and try really to go back and forth before the start of the December, I want to say. So, what is the main difference between a monoclonal and a polyclonal antibody? This is a very important step in every experiment you might do or application of a protein-related assay you will be doing or analyzing or reading about in the future. So, monoclonal antibodies are produced in multiple B-cell clones, while polyclonal ones are produced in the single B-cell clones. Monoclonal antibodies target multiple epitopes, while polyclonal target single epitopes. Monoclonal antibodies are produced in single B-cell and polyclonal in multiple B-cell. And monoclonal antibodies are derived from one animal, while polyclonals are produced from another animal species. Many times when you get questions like this about 2 different components, you will see that you will have an answer that is confusing you and putting you in 2 different situations, and it's just a flip-flop. We are 21 students today. Please, I would like everybody to answer so that you can have an evaluation of yourself. It doesn't matter if you do it wrong. Today, we are just training. Awesome. 86% did it correct. Great. So monoclonal antibodies are produced by a single B-cell clone. We discussed this many times that we have to do it. We get the clones and we have to really make sure we are getting only one clone, while polyclonal antibodies are produced by multiple clones. Okay. Okay. Next question. What is the major application of monoclonal antibody? Okay, so I have to make this bigger too. Which of the following is a major application of monoclonal antibodies in medicine? You could use it in treating diseases. You could use it in cancer immunotherapy. You could use it in diagnostics of viral proteins. You can do all that, or you can't do any of that. Think about the exam. Mostly multiple choices should be answered within 30 to 45 seconds. Difficult ones, it should take 1 minute to 1 minute and a half. And when we perform the exam construction, if we can say it as a construction, then we take into consideration how many choices I'm giving you and how complex is the question. So based on that, we'll decide on the time. Okay, 17 answered, 18 answered. Please go ahead and answer. If you're confused, at least put which one is your top priority and which one is the second top priority in your notes. And then you can evaluate how close you were to that answer. Okay. So 2 more people, please. Can you go ahead and submit your answers? Yes, no, yes, no. Okay. All of the above. You can target autoimmune diseases. You can use it in cancer immunotherapy, and you can use it in diagnostic. Be careful. There's 15% who were off. And there was one person who was thinking that monoclonal antibody. Be careful. We mentioned very carefully that we don't use polyclonal antibodies in medicine. We use monoclonal antibodies. So the question is valid here. It could have been written in a way that which of the following is a major application of a polyclonal antibody in medicine? Then there should be none, because polyclonal antibodies cannot be used. And we discussed why, because of their specificity. Okay, third question about antibodies. Why are polyclonal antibody advantageous for detecting complex antigens? There is a question. Can polyclonal be used in diagnostic though? In research, yes, but not in medicine. Like you can't use it really in therapies for people in the clinic. But if you're testing something in the lab and you're doing experiments and you're trying to validate, yes, you can start with the polyclonal antibody, and then the next part of the project would be how to make out of this polyclonal antibody a good monoclonal antibody that you can use in therapy. Okay, because on the slide, I don't think it was specified. Okay. Sounds good. Now you have it clarified, Gary. So why polyclonal antibody are more advantageous for research in detecting complex antigens? Is it because they are cheaper and they are produced in a much cheaper manner compared to monoclonal antibodies, or they recognize multiple epitopes of an antigen, and therefore that increases the sensitivity? Or they are always more specific than the monoclonal antibody? Or they don't require animals to be produced, so we can produce them in cells, like the description I mentioned to you earlier. So 15 people answered. Please push yourself to answer in the next 10-15 seconds. Okay. 5 seconds, please. Okay, for those who are not answering the questions, please have a valid reason why you are not. So, correct answer is they recognize multiple epitopes on the antigen, and therefore you can increase the sensitivity. Be careful, I didn't say increase specificity, increase sensitivity. Because if you have more than one antibody bound to the same antigen, then it's more chance to detect that protein or detect those antibodies binding to that antigen. Be careful that, yes, for those who answered they are cheaper, yes, they are cheaper, but they are not advantageous because they just are cheaper. The advantageous thing we are discussing here is detecting complex antigens. So it's not about budget, how much money you have, and what is the choice between monoclonal or polyclonal. Your logic is okay, correct, but it's about the sensitivity of complex antigens. And they are always more specific than monoclonal antibody. That's not correct. They are like less specific because monoclonal antibodies are more specific than polyclonal antibodies. Good. True or false questions about the RNA-seq. RNA-seq can provide information about both coding and non-coding RNA. True or false? Question. Not sure if it's just on my side, but your screen is frozen on the second question. Okay, so my screen is frozen apparently. I'm gonna go back. Sometimes this happens when I minimize or maximize. So how about now? I just went back to the original because when I maximize it, sometimes it freezes. Can you see it? Okay, great. So for RNA-seq, can you I'm just hoping everything. Now we see question 4. You see question, yes, now you see question number 4. RNA-seq can provide information about both coding and non-coding RNA. Is that true or false? 22 out of 24. Wow, we have some more visitors here. That's great. And the answer is, so happy that 91% is answering correct. For those who answered false, please note that RNA-seq is unbiased. RNA-seq takes all the RNA sample. It can be microRNA, long non-coding RNA, coding RNA, messenger RNA, everything. And it's mentioned in the slides. So be careful that this answer. Now, in the exam, if this question was posted, it could have been a multiple choice. Again, for RNA-seq, and please, if you don't see the screen of question number 5 now, let me know. It doesn't require converting RNA to complementary cDNA. If you are performing RNA-seq, you don't need to do that. 24 people, we are almost half an hour. Few more seconds, please. RNA-seq does not require converting RNA to complementary cDNA. Let's see who went through the material very extensively. False. Now, this is a very interesting result here because it's the first question where we get almost half of you divided into parts. Some of you said it's true and others, the majority said false. For those who said false, they have the logic that when you isolate the RNA, at certain point, you would need to make the complementary cDNA in order to be able to sequence it, right? For those who answered true, you have to be careful. And I would love if someone of you can give me the logic why you said it's true. Anyone wants to write or, oh, let's go to, yeah, so probably. Okay. Okay, that's fine. Okay, so the answer is you require generating the cDNA no matter what. Now, it's different when this complementary cDNA is being generated. So, for example, if I can make this clearer for you, if you are doing a cDNA library, the cDNA library has to be generated by taking that RNA, converting it to the complementary cDNA, and then making the double-stranded of that cDNA, and then clone it to a vector. But when you are performing RNA-seq, the steps are different, right? The steps, you isolate the RNA, you could make the cDNA and fragment it, or you can fragment the RNA, depends on the type. We discussed it, 2 different types. But then the way we do the RNA sequencing, remember that video that I showed you on the Illumina, where the first step is to immobilize the RNA into the flow cell. And then you have to generate a secondary complement. Remember the visual aspect. All the time I'm giving you the videos in such a situation. Try to remember the videos visually. Remember that it was generated, the first strand, it was washed away, then it's another strand that is washed away. So the bottom line, this was a confusing question, and you don't get too many of these in the exam, but there should be a couple or 3 questions like this to differentiate between those who can integrate the material and the information in the slides, plus the logic behind it. Okay. So RNA-seq cannot be used to identify single nucleotide polymorphism. Single nucleotide polymorphism, if you forgot it, it's only a change in only one nucleotide. And that only one nucleotide can generate a different RNA transcript, and therefore can generate a different protein transcript. Let me close this one. Oops, sorry. I mean, you saw the answer. Anyways, the idea is that, sorry, I was meant to click here, but my mouse clicked there. Great that 9 of you already answered, and it was 100%, and this is great. But yes, RNA-seq can be used to detect the single nucleotide polymorphism, because in principle, we can sequence every single RNA molecule, and we can detect the difference between every single RNA transcript. So why not? Okay, that's good. This is already half an hour of the lecture, and I hope this gives you a little bit practice. We will continue doing this in the next couple of lectures as well. And that explains why the amount of material you are getting is not really much, because we are spending more practice for the exam, and that's fair. The idea here is, and I'm keep repeating myself many times, this exam is not the end of your journey. I don't want you to fail. I want you to succeed. At the same time, it should be fair to everyone, for those who studied and worked, and for those who are new and they have no idea about the subject or have never took genetics or molecular biology before. I'm trying to be in the border between all of you guys, and it's really difficult with the number of the students I have in the class. The bottom line is, I wish you all the best, and I'm here just to support you. Don't ever think I am on the other side trying to act the professor who is trying to treat you bad. And I'm not going to take the part as a parent and tell you, guys, you have to study, guys, you have to be responsible. You know what you have to do. Sounds good. So we end this discussion about the exams and everything, and we just move to the PowerPoint. I hope you are seeing my PowerPoint now. If not, please say no. Or say yes, you can see it.
We can see it.
Okay, awesome. Awesome. So this is the third lecture in terms of the protein analysis, and now we are getting closer to the applications of what we can do if we have cells or tissues from different sources, and we eventually would like to analyze the proteins in order to get an idea if this person is healthy, is person infected, is there a reason that we could think that this protein is causing that disease, stuff like that. So what I'm telling you here is a snapshot of the protein analysis, but keep in mind always we can't do all this without the study of the background at the level of the RNA, the genomics, and the transcriptomics, because that can give us a lot of ideas. Yet the diversity that we get with the protein analysis, protein transcripts, and protein interaction, protein RNA interaction, protein DNA interaction is really wide. And since proteins are the vital molecules that perform most of the functions, so if we are speaking about DNA replication, proteins would perform that process. Polymerases are proteins. The same for RNA, the transcription process, the same for the protein function itself. It has to communicate with proteins. It has to communicate with macromolecules like lipids, like sugars, and that's why it has a central role in the cellular functions. Now, I stressed a lot about the antibody function and structure because without those antibodies or this class of proteins, probably the protein analysis setup, it would have been much more difficult, and at the same time, it would not be able to identify those functions. So I hope my slide works. Okay. So today's analysis is going to follow up on the immunoblotting, and we will discuss about some of the applications of the immunoblotting. I'm introducing you in each session to a little bit more technical aspects because those techniques are really difficult and it takes, I don't want to say weeks to months to master how to do those techniques and master how to get information that is useful from those techniques. The end of the lecture, we will be discussing the immunoprecipitation. So we started with immunoblotting last time, the immunoassays, and the immunoassays, and we will discuss the immunoblotting, how we get to do the immunoblottings, and in other words, how we can first isolate a specific protein and blot that protein into a membrane and then visualize it. So that's what we do in immunoblotting. But then one of the applications of immunoblotting is immunoprecipitation. So if there are many proteins in a cell, let's say, take an example of a cancer or two types of cancers, and you are assuming that the protein MYC, which is normally mutated in cancer, has other protein partners that perform certain function in one cancer versus another. You would like to know if MYC binds to that other protein partner. So this is how we can identify it by immunoprecipitation. Now, in the next lecture, I will describe for you how we can isolate cells based on their surface membranes by fluorescence-activated cell sorting, or is known as FACS. So recall, for those who didn't watch the previous class or were not present, recall that we always decide to look at a specific epitope after a lot of biochemical analysis, transcriptomics, and genomics that ended up leading to a sequence of a protein for a few amino acids that represents that protein, and we can specifically then identify that protein if we can find an antibody. That small epitope is recognized by the antibody. And in the beginning of this lecture, you can see I posted for you again the antibody structure. I went a couple of times on that, maybe quick, but you should remember that the antibody binds the antigen on these sites here. So these two sites and these two sites will be recognizing the antigen or that epitope of the antibody, of the antigen, sorry. If you look at the structure of the antibody, it is made of one chain. We call it heavy chain because it's bigger. And it is composed of two identical heavy chains. And these heavy chains are attached by disulfide bridges. Then you will have the light chains. And these light chains are smaller in size in terms of the molecular weight. And then those are attached to the heavy chain by disulfide bridges. So in principle, the antibody is a complex protein that is made of two different proteins. One is the light chain, and one is the heavy chain in duplicates. And those all together are attached. When I mention in duplicates, meaning it has two arms that are identical. And these two complex hinges are in fact attached by disulfide bridges. So disulfide bridges is something you learned in biochemistry and in organic chemistry, that's how molecules interact to each other. And you would see down the road that at certain moments in the analysis, we can break these disulfide bridges, and therefore we can get different visualization of an antibody. Okay. So then we discussed three different types of antibodies: the monoclonal, polyclonal, and the genetically engineered recombinant antibodies. How are they made and what are their applications? And we mentioned that nowadays we have a lot of ways to label an antibody at the complex terminal, where it normally complements bind to the antibodies within the immune response. This could be a fluorophore, therefore it can emit light. This can be an enzyme, HRP, horse radish peroxidase. It can change a substrate that is invisible into some substrate that has a color that we can detect. And also we can attach molecules which could allow us bind the antibody with other proteins. So there are others that I didn't describe for you because these are probably the three most immediate applications that you will get to. And in the next lecture, I will explain to you a little bit about the microscopy and the FACS and how the fluorophores are being utilized and what are the concepts behind that. But the bottom line is that all the molecular biology of the antibody and the antigens are based on the fact that we have an antigen, we study it a lot, which is a protein of interest. We decide to find with the bioinformatics, with transcriptomics, genomics, and DNA analysis, RNA analysis, and protein sequence analysis that we get to find a specific epitope that differentiate this protein from any other protein in the cell. To that epitope, we generate antibodies. In this case, that antibody would be primary directly binding to that epitope. And most of the techniques that we use is that that primary antibody is detected by another antibody. In this condition, we call it secondary antibody. And the secondary antibodies are mostly the ones that are conjugated, as I showed you here, so that we can detect how much of this antibody or how much of these two antibodies bind to each other. And indirectly, it's telling us how much of that protein is present. Then we discussed about immunoassays. We discussed about the ELISA, the types of the ELISA. In this image, you have the sandwich ELISA. So this antibody here, the capture antibody, recognizes this protein. And there's another detection antibody that recognizes another epitope on the same protein. And then we have the secondary antibody here, with the conjugation. And the more you will have of this complex here, meaning you are specifically capturing this protein, then the more signal you will get. And if you remember, we discussed about the ELISA plates, how they look like. And in this image here, the more the blue, the more you are detecting this specific antibody. And we discussed that there are more than one type of ELISA, depending on the experiment and depending on the types of antibodies available. All good. Now, how can I read the results that I see on a plate after performing ELISA? Well, just to be fair with you, this is not the right question. In fact, how would I set up an ELISA? This is the right question. So if we are discussing about a protein of interest, whatever that protein is it in cells, is it in blood, is it in tissues, wherever that protein is being available, then we have to make sure, if we are performing ELISA, to first have that protein being isolated by a certain company or in the lab, and we need to know how much of that protein is available, and we need to perform what we called a standard curve. So the standard curve, if I give you an example, tumor necrosis factor alpha, TNF alpha, which is an inflammatory gene, or any of the interleukins that are used in the immune responses, we need to know, or we need to have already isolated pure interleukin or TNF alpha, and we know the concentration. So we know that it's 500 picogram per ml. And then we would perform serial dilution of that. So it's 1 to 2. So the dilution is going to generate 250 picogram, 125, 62, 31, 15, 7, and 3. So you have multiple concentrations. I know how much the concentration in each of these tubes. Therefore, this is my standard curve preparation. In parallel, I have to have a negative control or background control where you don't have any proteins, 0 picogram of any proteins. This is normally just the buffers you use for the ELISA. And then you would place on duplicate the non-diluted 1-to-1, diluted. Each of these you will load it on in the plate in duplicate. And therefore, you can easily see now that the more the concentrated the standard curve, the darker the color you are detecting, right? You would perform the experiment exactly as I described here. So first, let's take it's this type of ELISA, indirect ELISA, let's say, indirect ELISA. So we will have first the proteins of interest added into the plate. And side by side, we will have the first 2 rows as a standard curve and probably the last one here as the background. And then all these samples here are from patients or from cells that you want to test how much of that protein is available, if this is interleukin, let's say. And so you will do the incubation, so the proteins are attached to the plate, and then you will put the first antibody directed to the TNF alpha or interleukin, and then after that you will wash, then you will add the secondary antibody, and that secondary antibody will give you this, when you add the substrate, is going to give you this blue color. And then if we are comparing healthy subjects to patients, all we need to do is to read this plate under the spectrophotometer. And the spectrophotometer is going to read how much is the so the diffraction of light based on the color of the product that you have. The more the blue, probably the more the diffraction, the less the blue is the less the diffraction, and therefore it gives you an optical density. So you have on one side optical density for each of those samples, including the samples in question. And on the other side, you have the concentrations. Now, for the healthy subject and for the patients, you don't know how much is the concentrations. But for the standard curve, you know. So you already know that these two wells, they have a concentration of, let's say, 500 nanograms, picograms. And each of these points of the standard curve, you know. So once you have more than 5 points that are consistent and this curve is great, and the R square of this curve is more than 90, then you can estimate based on the OD whatever concentration you like. So if we take the healthy subject here, and the optical density, let's say it lies there where I put my cursor on the screen, all I need to do is to make the y equal ax plus b on this curve, and I will directly know the concentration. So that's how I would know how much the concentration in each of these healthy subjects. I would look what is the optical density, and I will automatically use the curve to tell me what is the concentration. The same thing for the patient. And the most critical step is that you have a really good standard curve. If your points are not really good and the R square of this curve is not good, then the equation of the graph is not great, and then the concentration is not great. So this is basically the first and the most important step you have to perform in ELISA every time. Every time you run it, you have to do that. And we can use it also to get the concentration of some proteins. I will show you in the next couple of slides. But now, how do we use ELISA in health biosciences? I listed some of the applications that ELISA can be used. I mentioned before that if you want to detect a biomarker that is specifically expressed in cancer cells compared to healthy cells, all you need is just an antibody toward that marker, like MYC or RAS protein, those proteins that are affected, or a breast cancer related protein, and then you will perform ELISA and you can detect the cancer before even it becomes a tumor. Now, the other thing is you could detect antibodies that is used in autoimmune responses, because when your body produces antibodies toward your own tissue, you can take blood samples. Like this is an example for the lupus. If someone has that antibody, we know right now what is the right antigen to pick the lupus antibody. So we have antibodies against that. All we need is just to take blood from that person, and then we would look if there are circulating antibodies in his blood, and therefore he would be having this autoimmune response, similar to arthritis and others. You could detect the drugs like cannabis and cocaine and stuff like that. There are ELISA kits that you can detect those in urine. And also, if you are using a drug that you are giving to people to treat people against certain diseases, then you can try to detect if that drug is causing the generation of novel antibodies in conditions such as rheumatoid arthritis or inflammatory bowel disease. These antibodies, we already know them, and we can use them for that. Pregnancy tests, not the one that you do get from the over-the-counter. The one that you perform with your doctor after there are signs of pregnancy. These are also ELISAs. These are very specific to a protein that is the human gonadotropin protein. And I'm sure you have discussed this in the pathophysiology, because this is one of the earliest hormones that is upregulated during the pregnancy. If there are certain viruses that infects the body and antibodies are generated, HIV virus is an example. There is an antibody against the human serum cystatin C. Cystatin C. This one detects the infection of the HIV. And finally, the food allergen. So there's a lot of applications that the ELISA is being used, and it's a very sensitive technique. You can go up to the picogram. Picogram is like really very, very minute. And when I discussed with you about the ELISA, I mentioned that these are soluble proteins. So any soluble protein can be detected by ELISA. It's more practical for those proteins that are soluble in the blood because it's direct. You take the blood and you do it. It doesn't necessarily mean that you can do it only in the blood. If you have cells in culture or you have tissues, you can take the cells or the tissues, break them and get the soluble protein, and you can use ELISA as a method for the detection. Now, for the second type of analysis, it would be that we would need to probably look at more characteristics of the proteins. And there are three different aspects that I will be describing for you in this lecture and in the next lecture. It could be that we are just separating the proteins based on their molecular weight. And then we are staining for those. So this would be the ponzo. And as you can see here, this is the pink color. The more it's unspecifically binding to the protein, the more the protein is present, the more the dense the band are. And the bands are. And then the Comasi blue, again, you can see from the name, it's a blue color that binds unspecifically to the proteins, but this we do it in gels, in the SDS gels. You can also use silver staining, and the silver staining is more sensitive than the Comasi blue. So that's one type of analysis we can do. But for doing this, we have to first isolate the proteins, then dose the proteins. So we know that we are loading the same amount of proteins from, let's say this is patient 1, patient 2, patient 3, patient 4, compared to healthy 1, healthy 2, healthy 3, healthy 4. You have to have the same amount of proteins loaded in order to be able to compare between the samples. And I will show you how you do that. Here, there is a step where it's the immunoblotting. So the image you see, you have already immunoblotted the proteins into a membrane. So I will be describing for you how is that done. Now, in between, we can radioactively label proteins, and the radioactivity can be detected. So if we are looking on the accumulation after time of a certain protein, all we need to see how much radioactive material is being accumulated, because some of these amino acids are labeled. I'm not going to go so deep into this one. I'm going to discuss this one and this one for you. And they are kind of part of each other, so I will go stepwise. The first step, as I said, is to determine the protein concentrations. So let's imagine you have 10 patients and 10 healthy subjects, and you want to detect a cancer-related protein or tumor-related protein or inflammation-related protein. The first thing is you will take the same amount of protein from all of them in order to look for that particular protein of interest. You can't take 50 microgram from one person and 20 microgram from another person because you are already taking different material, right? So the idea is it's 20 microgram for everyone, 30 microgram for everyone, 50 microgram for everyone. So I want to know how can I make the concentrations of these samples similar, or at least to identify how much is the concentration in each sample. It's the same way that we discussed about making the standard curve here, but we are going to do it in a different way there. So here we will have first, if we have 10 samples or 5 samples or whatever, here we have 9 samples. You can see 6 samples, let's say, unknown. So I will take, let's say, 10 microliter of each of those, and I will add one of the 3 unspecific dyes that can be used to detect the proteins. I give you an example here, the Bradford. So there are 3 of them. There's the Lori, the Bradford, and the BCA. Okay. All of them work more or less similarly. They have a little bit less or more sensitivity compared to the other, but the idea is exactly the same. You will take the proteins of interest from which sample. You will take the same volume, 10 microliter, 20 microliter, and then you will add the reagent, which is the Bradford in this case, which is listed in this image as well. Now, the Bradford is depending on the Comasi blue reagent G250, which is unspecific and binds to the protein and gives a blue color. Okay, now, when the dye is not bound to the proteins, it's red. You can see it's kind of the less blue it is, therefore the less amount of proteins you have. Then when it binds to the proteins, it will give you a blue color. And then you can measure by spectrophotometer how much the optical density on each of these. And therefore you will be able to say, oh, this optical density is 0.5. Therefore, how can I get the concentration? In order to get that concentration, you need to make a standard curve again. But the standard curve here is not like necessarily to be interleukin-1. The most useful one is bovine serum albumin, BSA. I wrote it somewhere, probably it's missed in the slide. I will update the slide. So you would take bovine serum albumin and you make a serial dilution of it. Nothing, 500 nanograms or picograms, 1,000 picogram of BSA, and then 150 and 2,000. Then what will happen, you will get the standard curve again, because the BSA here, if you take 1, 2, 3, 4, and 5, and this is 1, 2, 3, 4, 5, and 6, probably there's one that is missing here, then each one of these concentrations, it has an optical density, and therefore you create the curve again. So you know what is the OD for the 500, you know the OD of the 1,000, the OD for 1,500, and so. Then your unknown sample, all you need to do is again to match it. And now you know how much proteins you have in each 10 microliters or in each microliter, and then you can select the same amount of proteins to be used in the ELISA analysis. Okay? Now, in this condition here, there are some aspects you can take into consideration. Probably the best way is to make stocks of your solutions of similar concentrations. But the idea is, if you don't know the concentrations, first you have to make the standard curve, get the concentrations, and then you go to perform the ELISA. Then you go to perform the ELISA or the Western blot. So this is the first step. Because maybe the 10 microliters here has 500 picogram, and the 10 microliters here, it has only 150 picogram, because the color is different already, right? So this unknown has more concentration than that unknown. So this step is very, very critical. You have a standard curve you make with the BSA, not the BCA. Be careful, the bovine serum albumin. You get the curve, and then you put your unknowns, exact same volumes, and then you can get the concentrations. After this step, you know how much you can use as a volume from each sample, and then you start to have the same concentrations in sample 1, sample 2, sample 3, sample 4, all the way. So this is the first aspect. The second aspect is visualization of the proteins on the electrophoresis. Meaning I have to separate the proteins based on their polarity. You all know that amino acids are different polarity. And therefore, if they are different polarity, this means different charges. And if they are different charges, then they can be repelled or attracted toward one of the cathodes or the anodes. The more positive the proteins are, they will be repelled again from the cathode toward the anode. And there's another point that I want to come in through the next few slides, which is the isoelectric point. It is the pH at which a specific protein has a neutral or negative or no charge. So for every single protein, there is a pH. If you play with that pH up or down, you will get to a point where this protein will become neutral. This is what we call the isoelectric point. Okay, so this is important sometime to isolate different types of proteins. Now, in addition to the molecular aspects of the charges on the amino acids and the sequences, then there are also the secondary structure and the post-translational structures, like I mentioned here, the phosphates. The phosphates are negatively charged. The more they have, the more the protein will be negative, right? So this is also taken into consideration when we are looking into protein analysis. So in terms of electrophoresis, you remember we discussed this for the DNA. It was much simpler because the DNA are negatively charged per se, because these molecules, they have phosphate groups. Now, for the proteins, it's a little bit different. We have to make them negatively charged in order to be able to separate them. Okay? The principle of the electrophoresis is based on the fact that we can separate molecules based on their first, the nature of the gel we're using, and the second is how much mass and the charge on that molecule. And in other words, the more negative or positive, or the more the charge is on that molecule, it can run slower or faster based on how big it is. That's why the charge, which is negative or positive, plus the mass, which is big or small, can dictate how the proteins are separated. And now if I go back to this, it starts to make a little bit sense for you that these proteins are bigger in size because they didn't migrate all the way down here. And these proteins are smaller in size because they are charged negative and they are running all the way from the negative to the positive. The same thing on these membranes. Great. So we set the ground that this is the type of electrophoresis we used for the DNA. It's normally horizontal. We place the DNA there. It's negative charge. It's gonna directly go toward the positive charge, which is here. And at the end of the way, you will say, oh, this DNA is here, is bigger than the DNA if run a little bit more. Now, in the protein parts, we are not gonna do that. We're gonna change the aspects. It's not horizontal, it's gonna be vertical. And the most useful electrophoresis that everybody uses in the labs nowadays, it's called the polyacrylamide gels, or polyacrylamide gel electrophoresis, and it's represented as a page. Remember that the page can be used for small molecular weight DNA. And since the proteins are very small, that's why we use polyacrylamide, not the regular agarose gel. Don't get confused between these two things, huh? Polyacrylamide can be used for small DNA and proteins, and agarose gel is used only for the DNA, because if you put proteins there, they will go out because the pores are very big. Now, the principle of electrophoresis is kind of presented for you here. As I mentioned, the smaller it is and the more the charge it carries, the faster it migrates. So if this is positively charged and that's the negative anode, so it's going to migrate so quickly because it's small and having so much charge toward the negative. The same here, if you have a negative charge and small, it runs so fast toward the positive charge. Now, yet, if you compare this guy to that guy, they both have negative charge, two of them. But this is bigger. So now the size didn't allow it to migrate that fast. And now I can differentiate by a bigger size, molecular weight, with similar number of charges compared to smaller size with similar number of charges. Okay? So that's the major aspect of electrophoresis. And that's how we separate proteins based on their charges and molecular weights. Now, how do we do that? In order to be able to do that, we need the acrylamide and the bisacrylamide. And these two molecules, organic chemistry, you guys are better than me in that. If you add phosphate radicals, then they will make this mesh-like. The more I add of the acrylamide, bisacrylamide, the more concentrated and the smaller the pores. Therefore, it will be more difficult for the proteins to pass through and the separation will be better. So the lower percentage gels can be used for nucleic acid because they are big and isoelectric focusing that I will come back in a slide from now. But the higher percentage gels, like 10% and above, this is what we use for proteins because proteins are smaller than DNA. Of course, we can make a gradient. We can make 10%, 20%, 10, 12, 15% in the same gel. We are not going to discuss that, but if you see it somewhere, this is not something really not normal, or we commonly use it. So this table here shows you if you use 5% acrylamide, you can separate proteins of 1,000 kilodalton. And if you guys don't know this unit before, kilodaltons is the molecular weight unit that we use for proteins or amino acids. Now, the more you add the percentage of the acrylamide, the more the mesh will be condensed, and therefore you can start detect smaller proteins. 5 to 30 kilodalton, you can detect them with a 20% acrylamide. So this is inversely proportional to the size of the peptide or protein that you would like to detect. If you are detecting big proteins, it's better to go with the low molecular weight, low percentages. If your protein is between 5 to 30 kilodaltons, then you would go with a 20%. So this is something you need to know in advance before you do the experiment. And now this is presented for you here. So we make just gels. We take acrylamide and bisacrylamide with the phosphate and timid, and we add them into the cassette. We put comp here so that we can load the proteins. After that, this is the type of the representation you get. The proteins are on the top, and you have here the gel. The more concentrated the acrylamide, the more concentrated these lines here, and therefore the porous will be much difficult for those proteins to pass. Now, when I run the electrophoresis, meaning I apply the current from the negative to the positive, then the proteins start to migrate. Obviously, the high molecular weight proteins, they run much slower. Medium size run faster than high molecular weight, and the smallest protein run much faster. And that's what I exactly told you about this image here. High molecular weight proteins are up, medium-sized molecular proteins are here, and lower-sized molecular proteins are there, and the same in these two plots. Great. So now we have isolated some proteins. We dosed and know how much the concentration of those proteins, and then we loaded them on a gel, and we differentiate between them based on the molecular weight. But there is one last characteristic I want to add, that you have to know: how do we get a unified negative charge around these proteins so that they can run? So the bigger the protein, the bigger the probability that it has a negative charge, right? So how I can make this unified across all these types of sizes of proteins, the yellow, the green, and the pink. The idea here is to use the SDS. Remember, the SDS is one of the detergents that we discussed. The SDS stands for the sodium dilysyl sulfate, and it has a negative charge, so it's an anionic detergent. Its ability to bind to the protein and gives a unified negative charge. Almost every 4 amino acids, I want to say, are bound by one SDS molecule. And the more SDS bind to that protein, the more negative charge will be added. Now, this will give you a unique way to differentiate between two different molecular sizes of different proteins. In other words, if you have two unknowns here, this guy is 31 kilodalton almost, and this one is around 20 kilodalton. In other words, this is bound by the same amount of SDS proportional to its molecular weight, and this guy is bound by SDS proportional to its molecular weight. Now, SDS also has other functions, which is most probably, you know, it breaks the disulfide bridges. It breaks the tertiary structure of the proteins. We are not discussing this in this slide here. I'm just showing you how the charges are being distributed over the proteins. So the SDS linearizes and binds to the portion of the protein and gives a negative charge. So I explained that for you. Now this is inversely proportional to the molecular weight, which means the more you have it bound, the more the molecular weight of that protein. And finally, when you run these gels, we need to have a standard molecular weight protein that we know. So you can see that in this image, they wrote down 92 kilodalton, 68 kilodalton, 43 kilodalton, 31. And this is a standard ladder or known protein of known size. Those we will buy them. They come from companies. And when we load these proteins, we know that this is the size 1, 2. We have a map that we know it. So this is our reference to guess what is the molecular weight of this guy and guess what is the molecular weight of this guy. Now, in this situation here, it's just a page, meaning I prepared a gel, amide, acrylamide. I made the comps. I made the gel. There are 2 components of this gel. I will describe sometime later. I loaded the proteins from different samples. The first one, I loaded the molecular weights that I know, a mix of the molecular weights that I know here. And then I have the first unknown loaded there, and the second unknown loaded there. Then I applied electricity, the electric current, and the proteins migrate. So the bigger molecular weight, 92 kilodalton, migrate a little bit. However, the 14 kilodalton migrated all the way to the bottom of the page. Normally, this would be our reference to stop the experiment. When we see this low molecular weight marker is almost at the end of the page, we stop the experiment. We stop the running, in fact. And now I can guess the molecular weights of these 2 guys simply by looking and matching the molecular weights. So this is the page. And in the presence of the SDS offered me 2 things. First, the negative charge toward the proteins, and second, the linearization of this complex protein. It becomes linear. Okay? Be careful and remember this very well. Now, I can add more reagents to the page. And remember in the first slide, I described for you the antibody, which is 2 heavy chains and 2 light chains. Now, if I add reducing reagents like 2-mercaptoethanol or beta-mercaptoethanol, the disulfide bridges are broken. And if I take an example, the antibody, the disulfide bridges between the heavy chains are broken. So it gives me a molecular weight for the heavy chain around 55 kilodaltons here. And the light chains are going to be broken into those around 25 kilodaltons. So adding reducing reagents can break the disulfide bridges. It becomes more linear. And therefore, if there is a complex protein of 2 different subunits or 2 different proteins, like in the antibody aspect, you will start to have 2 different molecular weights. Now, if I put that protein with no reducing reagents, like no beta-mercaptoethanol, it's going to have the sum of these 2 molecular weights. Okay, you can see here it's intact. You can see the high heavy chain and the light chain. You will see only heavy and light chain in conditions of reducing reagent, and the total protein in condition of there's no reducing reagent. So these are the different types of the SDS. First, the page we discussed. First the page, then SDS page by adding the SDS to that, and then reducing reagent on top of it. So you can see this is how we, with time, we get evaluated in terms of the page. And later on the Western blotting. Now, the isoelectric focusing, remember I mentioned that every protein has a pH by which it can be neutral. Here there are 3 different proteins presented for you, and all you're doing here is just loading the proteins and then changing the pH. You are not running here the page yet. So this is the first step. You are just changing the, sorry, I meant to say, you are not only running the page, but you are running the page and changing the pH. So what would happen is when you sort these, you're going to sort out according to their isoelectric point. If the protein becomes neutral at pH of 9, then it cannot migrate anymore. As much as it's charged, it's going to be migrating. When it becomes neutral, it's stopping there. The same thing for the yellow and the green. Those, we put them all together and we simply apply the electric current, and we have the reference here for the pH. Those who are positive, they are running toward the negative. And those who are negative, they are running to the positive. And that's what happened here. The yellow are negatively charged, they are running toward the anode. The green are negatively charged, they are running to the anode. But on top of that, the yellow, its isoelectric point is around 5.5 because it only ran to the limit where it's 5.5. It has no more charges. It's neutral. It stopped there. The same thing for this guy. It ran to the 4 point something. And now it's neutral, no more running. And for this guy, it's positively charged. It ran toward the cathode. When it reached pH of 8, it becomes neutral. So that's its isoelectric focusing point. So this is a merge between isoelectric point, isoelectric focusing and the pH. So we can detect the charge of the proteins. We can detect if they have glycosylation or phosphorylation, because these guys will offer more or less charges on a protein. And then we can do 2 rounds the page. We can do the first round by running and separating according to their isoelectric points. And then the second round, we can separate how many proteins there are. I'm not going to describe this too much. It's a little bit complicated, but it's listed for you here. Okay, so how much time I have? Not even 1 minute. So I will stop at this slide and we complete in the next. I will stop recording right away. And if you have questions about what I said, stop sharing and stop recording.